It's a great pleasure to welcome you back. I'm very happy to introduce our first speaker, Jan Vijg from Albert Einstein University. I'm a great fan of his work. He's focusing a lot on sort of DNA damage and how DNA damage and mutations can contribute to aging. Are you there, Jan?
Yes.
Fantastic. And we can see your slides now, so everything is wonderful. So whenever you're ready, you can go ahead.
I am. All right. Well, I really apologize for not being able to find my favorite green icon, but we apparently found it back. I thank the organizers for inviting me. I really appreciate it. It's a wonderful meeting with all sorts of promises about living longer and living healthier. Now, in order to do that, let me get my poster. In order to do that, we actually need to know a little bit more about the black box that we call aging. In spite of almost a century of research, we know surprisingly little about basic mechanisms. There are, of course, hallmarks, but the question is if hallmarks are enough, because, of course, we will never know what are the primary and secondary causes. There's probably a hierarchy of changes that are happening during aging, and fixing hallmarks or fixing symptoms is not what you ultimately want to do. Now, half a century ago, there were two basic theories of aging. One is actually protein error catastrophe theory, and the other one is somatic mutation. So the argument would be that there would be an accumulation of somatic mutations in somatic cells, or an accumulation of protein errors, or both maybe, that would ultimately be responsible for aging as the ultimate cause. But the problem is that even after half a century, it has not been possible to test these hypotheses, mostly because of technological problems. The technology to do that is really not there. Both are stochastic theories, and we still have a lot of trouble in addressing these problems. I will mostly talk about somatic mutations because that's actually, I can say now, virtually resolved. Technical issues are resolved, and we are actually in the process of testing that. Now, what are somatic mutations? Well, most people, they mix up DNA damage with mutations, and I'm sure you heard about it at this conference multiple times. DNA damage is actually very frequent. The DNA molecule in your cells under physiological conditions is not very stable. There are probably 100,000 lesions per cell per day, and they have to be repaired, and they are. They're repaired extraordinarily quickly and efficiently, and also usually error-free, but there is still a very low error rate, and those are the mutations. Wrong base pairs are putting in, or there are small deletions, chromosomal aberrations, and those changes are actually irreversible. So DNA damage is reversible, but this as an endpoint, DNA mutations is not. And we, of course, know that mutations are actually essential. There are only two factors responsible for life as we know it. One is mutations. They have to generate the variation. And the second is natural selection. So they are really totally responsible for the diversity and also the continuation of life. So it would be really ironic if aging of somatic cells in multicellular organisms would actually also be caused by somatic mutations. Now, in multicellular organisms like humans, somatic mutations works as follows, of course. There's first the zygote. Actually, I indicated it cleanly, but it's not. There are probably at least 60 de novo mutations already in the zygote due to mutations in the germ cells. But you can see the pattern here. Mutations will accumulate gradually and randomly, different colors, different mutations. Occasionally, at a later life usually, there is a particular mutation in a normal cell that is selected for because it gives the cell a growth advantage, and you may actually get a tumor out of that. So you can actually take the tumor, sequence it, and then you know what the mutations were that were present in that original cell at that particular stage of life of that individual. But there are also other mutations that by drift or other reasons are expanded, clonally expanded. So this is the pattern that you see, a lot of random mutations and an increasing mutational burden from cell to cell that are all different. And sometimes you see clones, tumors or non-tumors. Now, it used to be very difficult to actually quantify that, but now we can. We have a number of approaches. I list the three most frequently used ones. First of all, you can take I mean, the problem here, again, as I pointed out, is that this is a stochastic process. So every cell is different. You will need to take a single cell approach. You can do that by expanding cells, mostly stem cells in culture. You can take cells like, for example, Maria Eriksson did very nice work on human muscle stem cells, and she made clones out of that, and she can then sequence the clones. But in this particular paper, they actually took organoids. So they come from a single stem cell, and then at the end they sequence the organoid, the clonal amplification. And then you can do a whole genome sequence. You can, of course, compare that to the Germline, and you can see if there are de novo somatic mutations. Now, an alternative approach is that you simply look for the small clones, non-tumor clones, that I already pointed out before. That's actually that you take a tissue like a liver, you take very tiny biopsies in a raster across a particular slide or so, and then you do bulk sequencing of these small pieces of sample, and at a very high depth, and that is when you find a clone. If there is a clone, like more than one cell, many cells that contain the same mutation, you can find it in this way, and that has been done. Both approaches have been taken, and it is very clear that mutations accumulate with age. We already knew that before from other technology, but there's absolutely zero doubt that actually happens in virtually all organs and tissues. Now, the problem with these two approaches is that you are dealing with either you're looking at stem cells, and I mean, mutations do accumulate in stem cells too, but maybe less so. And of course, you miss a lot. So the best way, the golden standard, is really to do true single cell, whole genome sequencing, which we developed by two people in my lab. And you have to compare with bulk DNA. This is an example from human B lymphocytes, different ages. You can see that in both bulk DNA and a single cell after amplification, you can see single nucleotide polymorphisms, because they, of course, are present in every cell when you compare it to the reference genome. But a de novo mutation can only be picked up in a single cell. You can see that here. We usually have an algorithm that checks the validity of this de novo sequence by linking it up to a SNP. Now, using this system, single cell, whole genome sequencing, after amplification, we observed that there was indeed an increase in somatic mutations as a function of age in these cells. You can see it goes up from a couple of hundred per cell to several thousands in very old people. There's also a variability from individual to individual and from cell to cell. You would expect that. It's a stochastic process. And also there's a variation from individual to individual. Again, we see that quite often during aging. Sometimes there are enormous outliers. So some cells have a very high mutation. It's rare, but it does happen. Now, in this particular case, we looked at mutation accumulation during aging in these B lymphocytes. We looked at the functional genome, and we looked at the genome overall. The functional genome is the part of the genome that has transcribed sequences, so the transcribed part of the exome, plus regulatory sequences, and there are many of it. So the total amount of the functional genome is about 10% of the whole genome for a particular differentiated cell. That's sort of a rule of fist. What you see here is very clearly that they accumulate much slower in this functional part of the genome than in the genome overall. That tells you that they must have a function. There's a functional loss somewhere. It gives the cell a disadvantage in doing the whole process that gave rise to these B lymphocytes, from stem cells to eventually further differentiation. You can see that they lose cells, they go by the wayside. In liver, the situation is still a little bit different. There's also the increase with age, again, from probably less than 1,000 mutations per cell to actually a lot in people of like in their 70s. We could not get, of course, so easily liver biopsies from centenarians. It's difficult. But when you compare these age groups between B lymphocytes and the liver here, that is about twice the level of mutation. So mutation accumulation in your liver is much faster and reaches much higher levels than in your B cells. Now, an interesting thing here is when we pick these individual biopsies, we also turn them into organoids, which of course we can do too. You can see that here, we do whole genome sequencing. We find that this is the mutation frequency. It's about 1,000, a little bit more than 1,000 per cell. If we pick cells from these organoids, single cells, and we use our single cell approach, it's exactly the same. So it tells you that the single cell approach works very well. But the interesting thing here is when you compare it to the single cell approach on differentiated hepatocytes, you see that on average, mutation frequency is far higher, but also the variation is far higher. So it's tempting to conclude, to say, of course, that what's happening here is there's a hierarchy of cells from stem cells, and they difference all the way to completely differentiated hepatocytes. And those are the cells that provide function. So it's really important to actually look at the differentiated cells. And you cannot do that using clonal outgrowths. You really need to take a single cell or single nucleus approach. There are, of course, also many tissues where you will not be able to actually see anything growing out, like think of human neurons, for example. Now, so stem cells then have a much lower mutation frequency, still accumulates with age, but not as fast. Now, if you look at germ cells, the germline versus the somatic mutation versus the soma. In this case, we did that, students in the lab, Brandon Milholland. And in humans, it's actually not so difficult to do because you can simply compare offspring and parents and basically count the number of de novo mutations that you see. But somatic, of course, and we did it in the mouse too, but somatic, we can now use our single cell approach. If we did that, and you can see that there's a surprisingly high difference. So this is mutations in the germline, and this is in the somatic cells. So there's almost 2 orders of magnitude larger, higher mutation frequency in the germline, which tells you that your germline is pretty well protected. And we already saw that it was true for stem cells as well. Now, if you look at yet another tissue, this is human lung, and we were lucky to have Simon Spivak, a clinician who was able to do bronchoscopy and get us these lung cells from these subjects. And you will see this is a little different than the previous ones. In the previous ones, I showed you the actual single cells. Here is the average of multiple single cells, the median number of mutations per individual. So multiple single cells per individual. But the effect is the same. You will see that in normal, in this case, non-smokers, the number of SNVs is really going up. And here we looked also at small indels, small insertions and deletions, and it's the same thing. Now, if you smoke, then you can clearly see that both your mutation burden is higher and the rate at which your mutations accumulate with age is higher than in non-smokers. Now, if you're interested in looking at these somatic mutations in normal human tissues during aging, go to our new database where this has not come out yet, but it will hopefully soon. And here we sort of collected all the somatic mutation information that's present now in human tissues during aging. There's an awful lot of that, so it's really interesting. So that's why I know that we will have now finally tested this very old hypothesis of aging very, very soon. Now, the problem is, of course, what does it really mean? You notice that the number of mutations is not small. Only these base substitutions, where you're talking about thousands in an old person per cell. So it would be interesting to see what happens when you look at a possible relationship with longevity, with lifespan. And we could do that thanks to Vera Gorbunova, the next speaker, who studied all these rodent species from naked mole rat, guinea pig to mouse. This is very expensive, so we couldn't do many more than those. But we did a limited number, and when you do that, and you take cells, in this case, primary fibroblasts, lung fibroblasts, that Vera took for us from these species, then you see that only the mouse has a significantly higher spontaneous mutation frequency. The other species, the cells in the other species doesn't really show that. But if you now give them a challenge, you take these cells and you challenge them with different doses of bleomycin, which it's a classigen, but it also induces point mutations. Then you will see that the mouse is still uniquely high. Mutations are also induced in the mouse at a fairly high level. And this is the lowest dose of bleomycin. Any higher dose and the cells die. So mouse cells are also very susceptible to the cell killing effects of this mutagen. But if you now look at the other species, there is a longevity relationship. Guinea pig, which probably lives 14 years, you will also see that when you look at the dose response, there's a rather robust increase in mutations. In black mole rat, I believe it lives 25 years, it's less than that. And in naked mole rat, it's very low, very similar actually as in humans. So there is this idea that the mutation burden is actually correlated with the longevity, or the shortevity, I should say, because it's higher in shorter lived animals like the mouse. Now, for the mouse, this may actually be an exceptional case, because we notice that this polymerase eta is different when you compare the mouse to the other species, and this may actually be responsible for the fact that this is so much different from all the other species. But generally, there is such a longevity relationships. And very recently, I got an email from Inigo Martin Corina, who has a paper on bioarchives right now, who did this in many more species, not only rodent species, it's all other species as well. And he took a little different approach, but he also confirms that there clearly is such a relationship. It's the first time that that has actually been demonstrated. Now, how could it work? How could somatic mutations cause aging or age-related diseases? There are essentially three mechanisms, and the first one is very simple. Of course, we have thousands and thousands of genetic diseases caused by a mutation in the germline. But what most people don't know is that there is also a somatic mutational variance of probably all of these diseases. When there's a mutation, probably very early in the embryo, then a substantial fraction of your adult tissues contain that mutation. And when that mutation is in this disease gene, then that can actually cause a disease. The most spectacular case was Alzheimer's disease, when there was this early somatic mutation in the presenilin gene, and that actually gave rise to Alzheimer's disease. When you go a little further in age and you follow the aging process, then it could still be that cells emerge with a de novo mutation, like these two here, that will at old age grow into a clone. They begin to grow and they're clonally amplified because there's an advantage. I told you that already earlier. That is, for example, a tumor. But not only tumors, there are lots of other clones that are now discovered, and we don't really know what they do. But that could definitely contribute to aging. Now, finally, and in my opinion, the most interesting thing is it possible that just an increased burden of somatic mutations randomly, but beginning to affect, of course, all your gene regulatory networks, can that actually lead to decline of function? Well, obviously, we don't really know that. That's something we need to figure out. But it's possible that that actually happens, that this increased somatic mutation burden could begin to affect transcription. It will lead to increased transcriptional instability. You will see a sort of a random variation. While initially, this is what he showed a long time ago, this is the mouse, cardiomyocytes of the mouse. Initially, of course, when you look at a particular gene, the transcript level is the same from cell to cell. It's supposed to do that. It's supposed to provide function in a correct manner. But we notice that at older age, that's no longer the case. Cells begin to randomly vary their transcript levels. And that could lead to loss of cell fate in certain cases. That was demonstrated by Steve Quake's group a while back. Actually, this took a long time before people began to confirm this, but now it's generally accepted that there is a quite significant increase in this transcriptional noise, as we call it. There are multiple papers on that, on the multiple cell types. Now, conclusions then for the moment is, first of all, that somatic mutations definitely accumulate with age. We know that for a fact for virtually all organs and tissues. And they do that at a tissue-specific rate. I just showed you that liver is higher than B lymphocytes. Actually, lung is a little bit higher than B lymphocytes too. And others have found the same differences. Now, there's also differences in somatic mutation rates depending on the species. I showed you that there's probably a correlation now with species longevity and the mutation rate. But also environmental factors are important. I showed you smoking, but there are others as well. And the character of the cell that you're looking at, stem cells are more resistant to mutation accumulation than fully differentiated cells. Germ cells are very well protected. We also showed that somatic mutations have fitness effect. Others have shown that too. Marie Eriksson, for example, found a correlation between the mutation burden and the capacity of those cells to actually grow out into clones. And finally, of course, it must have occurred to you that somatic mutation rate, the burden that you detect in, for example, your blood cells, could actually be a risk factor. It could tell you how rapidly you will develop cancer or maybe other diseases, and we have found some evidence for that in smokers. I had no time to actually show that particular result. Now, what are we going to do now? Well, of course, we only focus on one thing, and that's causality. Is it true that you find high mutation burdens, and do they cause known phenotypes of aging? That's obviously the next step that we are going to take. Now, for the sake of this conference, can we do something about it? I used to say no, but I'm a pessimist in that sense. And actually, there are a number of approaches that you can take to even fight randomly accumulating mutations, possibly also randomly accumulating protein errors. Now, there are preventative measures, and that's very obvious. I mean, don't smoke. That's always smart. Don't lay in the sun. You get mutations, lots of them, and you actually get cancer. Now, there are also dietary anti-mutagens. I'm far from an expert there, but there's a lot of literature evidence for such compounds. So you can do something in a dietary sense. There are also metabolic interventions that you can take. We demonstrated already a couple of years ago that mice subject to dietary restriction do indeed have lower somatic mutation frequencies. Also, we showed a little bit more recently that FOXO, when you overexpress it, then the mutation frequency is lower. And when you suppress it, downregulate FOXO, then actually the mutation frequency is higher. So there may be ways here to do that. You could obviously think of upregulating DNA repair pathways, but I learned from Jan Hurmakers, and I of course agree with him, that that's actually extraordinarily difficult and probably highly unlikely to happen, but you never know, and we should try. And finally, and I think that really is an option, a viable option, is to try to eliminate mutant cells and mutant clones, try to identify the target, the possible therapeutic targets for that, to eliminate those cells and those tissues. So you always keep a healthy tissue. These are the people who did the work, and I already showed all their pictures to you and told you, and our collaborators. Thank you.
Thank you so much, Jan. Wonderful, wonderful talk, Jan. We have time for one question, and the one that's most upregulated is my own question, so I'm very happy about that. Loss of mismatch DNA repair increases mutational load, so I'm talking about Lynch syndrome patients, leading to greatly increased risk of certain cancers. But these patients don't develop accelerated aging. Do you have any explanation for these observations?
Yeah, well, first of all, we don't really know, I think, if they don't develop accelerated aging, because they usually die from cancer very early on. But it's also possible that it depends on the type of mutations. In this particular case, you see an accumulation only of point mutations. So there are many other mutations. I had no real time to go into that. I briefly mentioned small indels, but there are, of course, lots of much larger genome rearrangements, and they generally have a much larger phenotypic effect. So that could be one of the reasons. But overall, I really don't think that we know that these mismatch repair deficient mice do not develop proteroid symptoms. I checked that with people here at Einstein who do lots of work on them, and they don't know either. They never studied them for premature aging.
Okay. Thank you so much, Jan. There are questions on Slack. It would be great if you go there, including questions on line elements, which I guess could be some larger rearrangements. Let's give Jan another round of applause.